Be here. My lab is very interested in understanding the phenotypes that are associated with aging and human aging, and understanding the molecular basis of these phenotypes. So there's a long list of phenotypes. So the most prevalent is graying of hair, and there's been a lot of discussion about biomarkers of aging, and you could consider the graying of hair as maybe the best biomarker of aging if we really want to affect aging. Graying of hair is the most prevalent feature, so we should be able to affect graying of hair so that's one aspect. Of course, there are other features that are also very prevalent. So 40% of everybody will develop some type of cancer, and there's other things, osteoporosis and dementia is between 10 and 20%. So there's a long list of features. This is just a small list. It's based on downloading PubMed basically and looking for aging features, but there's obviously many more. So it's a very complex phenotype, aging. So we've also looked at this in pathological samples. So we looked at 33 million histological samples and looked at the text description of these samples. I'm not going to talk so much about this study today, but you can go on bioRxiv and read about it. Hopefully it will soon be published, and this is led by our text mining expert, Michael Ben-Ezra. So we use these type of analyses where we can quantify phenotypes in different diseases, so monogenic diseases typically. So this is discotosis congenita. This is a disease where you have issues with your telomeres, and these patients develop pigmentation changes, which is an age-associated feature in humans. And then they also, eventually they have bone marrow failure, which is what eventually kills them unless they get bone marrow transplantation. But many of the features are not so aging associated. 16% of the patients develop graying of hair. So there's a wide variety in how the phenotype of these patients, and it sort of indicates whether or not telomeres are so important in human aging. So Lynch syndrome, I think, is also an interesting one. This is based on data from the registry, and this is a disease where you have defects in mismatched DNA repair. So these patients develop a very large amount of mutations in the genomes, and they don't actually develop premature aging, but they do develop cancer to a high degree. And so this indicates or questions whether or not mutations by themselves are really sort of causative in many of the phenotypes we see with aging. But because we have a quantified version of these diseases, we can then do different types of clustering, hierarchical clustering, for example, and look for how different phenotypes associate with each other. So here we can see short stature, microcephaly, and developmental delay form a cluster. There are other clusters here. This is a cancer cluster that associates with sun sensitivity. This is typically premature aging diseases where you have defects in nucleotide excision DNA repair. You also see neurological features that associate with each other. So this is interesting, but you can also see how different diseases associate with each other. And you can include aging and then see how they form a cluster and with which diseases aging most closely resemble. And so this is something that Daniela spoke about earlier, that we have used this aging phenome, this collection of phenotypes that we have in aging, to identify new genetic diseases or genetic diseases that have not previously been associated with premature aging. And so we screened these 5,000 some diseases in the OMIM database, and we identified this Woodhouse-Sakata syndrome. And so in the hierarchical cluster, Woodhouse-Sakata syndrome associates closely with mandibular dysplasia and progeroid-like features with lipodystrophy, MDPL, which is a POLD1 deficiency. It also associates with classic progeria, Hodgkin-Gilfus progeria, and Werner syndrome, as well as normal aging. The patients have premature aging. If we do facial photographs, we can see that they display accelerated aging using computational analyses. And we were fortunate, and this is actually an interesting story, so this is sometimes you get lucky in science. So there was an individual in the lab, his at that time girlfriend was working at the genetics department at the hospital, and they were talking one evening, and she was saying, like, I think we have those patients. So they actually had this very rare disorder They had 2 patients of that. They're the first in Northern Europe, and they have also display features of premature aging, looking at blood biomarkers. And so Daniela has already spoken about this, so I'm not going to say anything more about this other than the idea is that if you are unable to maintain a stable R-loop landscape, if your R-loop variations increase, you get premature aging. So DNA repair and DNA metabolism seems to be very important in aging. And so this is highlighting work from the great Dr. Garrick Magarattichan, who presented this idea that if we could figure out molecules that could stimulate DNA repair, that could elicit an adaptive response, then maybe we could, that could be helpful. So what does not kill me makes me stronger is sort of the term we all think of when it comes to hormesis, so this cellular adaptive response. And so in this project, we collaborated with Silico Medicine. I think you've heard about this company, amazing company. And we identified drugs that were scoring high as a possibility of stimulating DNA repair. I'm not going to talk so much about the project other than we identify molecules, for example, molecules. So these dots are molecules that are treated with Petri dishes with cells that are treated with different molecules. And so here at day 0, we hit them with 16 gray ionizing radiation, which is a high dose of X-ray. So we really hit them with this X-ray hammer, and then we can follow them over time. And we can see that some of these drugs that we treated these cells with make the cells like completely resistant to ionizing radiation. And so we did some more work. I'm not going to go into great detail, other than saying that we found drug F, which seemed to reduce DNA damage. So this is in replicative senescent cells. There's fewer of these 53 BP1 foci. This drug reduces markers of cellular senescence, beta-gal. It also increases lamin B1, and cells can grow for a longer period of time in culture. And what's remarkable about this is actually that you can damage cells, and the DNA damage is removed. And this damage is thought of to be persistent. This is work from Judith Campisi. But the damage actually gets removed, and the cells return into cycle for a few more cycles. So you can see that the cells start expressing pCNA, which is a marker of DNA replication. We can also see that predicted cellular senescence decreases, and I will get back to that a little bit later. So this is something that we're spinning out as Vanaheim Bio, and please get in touch with Garrick if you're interested in living healthier for a longer period of time, and if you have money that you can invest in a company. So this is one way that we're looking at aging. We also look at tissue phenotypes in aging. So in this project, we have tried to identify what happens with nuclear morphology, which is affected by DNA damage. So if you damage a cell, for example, with ionizing radiation, the cells become senescent, and the nucleus of the cell actually changes shape. So we thought that perhaps we could identify senescent cells based on their nuclear morphology. And so we did this using machine learning approaches. So this is, we have a segmentation algorithm that can identify the nucleus, and then we can train a predictor to predict whether or not a cell is senescent or not. And it actually works really well. This is in culture. You have 95% accuracy. It correlates quite well with different markers of senescence. And because we wanted to make this sort of more universal, we could do different types of normalization, so we can remove the background. We can also size normalize the cells, and then we can mask the internal regions. The reason we do that is because there's a large difference in how the nucleoli are found in different cell types. And when we do these types of normalization steps, we can see that the algorithm does lose a little bit of its predictive power for cells and for cells in culture, but it's still actually pretty good. And so we can predict more senescence in cells from premature aging diseases, and also in astrocytes, neurons, and in liver cells. And so, to our surprise, if you have less senescent cells, so this is in human skin biopsies, if you're down here, you have actually an increased risk of developing cancer in the future. So this is all published in a great journal called Nature Aging. We then wanted to investigate this further, and this is work that's unpublished but coming out very soon. So we got access to the Komen Tissue Bank. It was in collaboration with Chris Benz at Buck and Mark Powell. And we could then screen for senescent cells in these biopsies and then see if there was an association with future breast cancer risk. And so we can see that nuclei actually seem to cluster quite well in different tissue types. And this is a UMAP, and each dot is an individual, actually, so there's more than 4,000 individuals here. We can also do spatial senescence prediction, so the cells can, the idea was that perhaps you have foci of senescent cells. This has been shown before, and we also see that actually. So with the senescence predictor, we can see that if you have a senescent cells and look at the cells immediately surrounding the cell that's predicted to be senescent, those cells also seem to be predicted to be more senescent. And vice versa, if you have low senescence, you're actually also predicted to be low senescence. And this is in fat, but it also seems to be the case in other tissue compartments. What's important here is that we can then use the senescence predictor and combine that with other scoring algorithms, and then we can actually, with very high, we can risk stratify women to a very high degree. So if you have, in this case, low senescence in the adipose tissues with this predictor and high GALE, you get an odds ratio of 4.7. And so this means that we can potentially risk stratify women that have taken biopsies during mammography, and this is coming out in Lancet Digital Health very soon. But we have actually gone in now and looked at women that have undergone mammography where a tumor has been found, so benign tumor, and when you see that on mammography, you take a biopsy. Fortunately, the majority of those are benign, but we could potentially use that as a way to risk stratify, and indeed, we can see that we find an odds ratio of 2.5 for using our predictor. So that indicates that potentially we can risk stratify women this way. All right, we can also use this in more clinical settings. So I have two examples here. One is actually an interesting project that was led by, or is led by Amalon Teklu, who's Ethiopian and is in Ethiopia. And we were interested in hypoxia and health in this project. And Ethiopia is very interesting in this context because you have highlands and lowlands areas. It's actually the largest, I think, population in the world where you have this highland and lowland areas. So what we can see is that looking at age-adjusted DALYs, so this is the disability-adjusted life years, so the amount of disability you have, it actually decreases with elevation. So the higher altitude you're at, the less disability you have. So this is really interesting. We could also go in a specific region of Ethiopia called Tigray region to see if we could see a connection between hypoxia and senescence. And the way we could do this was because they're pretty good at doing blood smears in Ethiopia because they have a very high level of malaria. So we could do blood smears, blood draws, and then we could determine senescence in the blood smears by looking at nuclear morphology as we have done before. And we can see that some types of senescence is increased. The music started. We have to eat soon. IR senescence is actually decreasing. So DNA damage-induced senescence actually decreases with elevation, which could support this idea that oxidative DNA damage could contribute to some of these senescence features that we see. We have also run another clinical trial where we've tried to see if we can influence senescence. And we decided to investigate COPD. And this is, I guess, a main cause of death globally. And the reason that we're interested in this particular disease is because it's caused majorly by smoking. And when you smoke, you induce DNA damage. And so DNA damage can activate this enzyme called PARP1. And PARP1 creates polymers, pol-ADP ribose polymers, that leads to loss of NAD. And I've done quite a bit of work before investigating this area, looking at how PARP1 drives NAD loss and various phenotypes. So we know we can inhibit PARP1, but we can also use a precursor for NAD, nicotinamide riboside, and then increase NAD levels. So the idea here was to maybe use NAD precursors, because we know DNA damage is central in COPD, and inflammation may be driven by PARP. And so we did a double-blinded, placebo-controlled RCT, where we had 40 COPD patients that were randomized to NR placebo. And what we saw was that COPD patients in general had lower NAD levels, and the lung function actually significantly correlated with the amount of NAD in the blood. When we give the patients NR, we increase NAD levels. The increase in NAD is not dependent on the previous level of NAD, so they all seem to kind of get a really quite large increase. Our primary outcome measure was IL-8, and this is actually met. So patients on NR actually see a significant reduction in IL-8, both directly after the intervention, but also at follow-up. The reduction is to some extent dependent on the amount of starting IL-8, and this reduction also correlates then with a reduction in recruitment of neutrophils to the lung of the patients. We asked the patients to cough up sputum, which is like mucus that's in the lung, and then we could look at cells in the mucus using our senescence predictor. And we can see this is, it didn't reach significance, but you can see that there's a trend that IR senescence and regulative senescence is decreased in the lung of the patients. We also looked at DNA methylation clocks. We don't see any significant difference here. What we do see is there's a very strong significant association between IL-8 levels and Horvath's 2018 clock, and a very strong correlation between IR predicted senescence and also Horvath's clock. So this suggests maybe there is something there. We did transcriptomics, and we can see that there's a very strong sexual dimorphism actually in the nasal epithelium of these individuals. And we see that NR seems to reduce inflammatory components. And this is actually even stronger in the healthy controls. So this is coming out in Nature Aging very soon, and sort of indicates that maybe NR could be a way to alleviate inflammation in COPD. I want to highlight this, that this study, this is the budget. This is not something that you ever see it's because I was just told that clinical trials would cost $30 million to run. So this is a trial that I think has clinical meaningful outcomes that can be followed up. It's a small trial. It only costs $260,000 US. So we can run clinical trials. We can actually test some of these things that we have for a relatively low amount of money, okay? Fortunately, there is a number of trials that are underway in humans, and you can go in on this database and look at these trials. And with that, I'm just going to stop and thank my lab, my collaborators, my funding sources, and thank all of you for listening here before dinner. Thank you so much.
Thanks. Maybe we have time for one quick question.
One?
Yeah, it's late.
These very tough, these organizers.
Well, great, lots of great stories. So quick question, for your DNA repair activation, what is the strategy? Enzymatic activation, or do you increase expression of some DNA repair genes?
So we know actually the target, which is sort of not a canonical target of the DNA repair, sort of peripherally associated with the DNA repair machinery. I would say an understudied part of it. I cannot tell you that because then we cannot patent it, I think. And the CEO is going to be very angry. So already very angry. Yeah. So, but hopefully we have it will be patented and then we will disclose it.
Okay, I think we need to stop because we need to continue at 7 and we can have dinner now.
Thank you so much.